Hello everyone, welcome to the next lecture of microbiology. So today's topic is Pseudomonas and Vibrio. This topic is from the section of bacteriology. So both these bacteria are gram-negative bacilli and they have certain characteristics which are similar in nature, and that's why both these organisms are clubbed together. Both Pseudomonas and Vibrio are a large group of bacteria which can cause important clinical medical conditions, and you should remember that the important members in each of these two groups are: from Pseudomonas it is Pseudomonas aeruginosa, and from Vibrio it is Vibrio cholerae. So we will see what their characteristics are, and how they are grown in the lab, and what medical conditions they can cause, and what is their significance, and some other features. So first of all, we will go to the first section of today's topic, that is Pseudomonas. So as I already said, Pseudomonas is a large group of bacteria containing various different important species of this genus Pseudomonas. So first we will see the basic or simple classification of this Pseudomonas. So the family this Pseudomonas genus belongs to is Pseudomonidaceae, and the genus is Pseudomonas. There are various species which are important in this group, like Pseudomonas aeruginosa, Pseudomonas cepacis, Pseudomonas tutzeri, Pseudomonas putida, Pseudomonas fluorescens. You don't have to remember this classification, or you don't have to remember all these names, but this is just for the orientation, and you basically should know that Pseudomonas is a group of bacteria containing different important species, out of which Pseudomonas aeruginosa carries the most significant value in our medical studies of bacteria. So here we are seeing the importance of Pseudomonas. Majority of the Pseudomonas are saprophytic. That means they are found in environment, and they gain their nutrition from the organic material. So decay of the organic material is caused by bacteria, and Pseudomonas is one of those different bacteria. So it is found in soil, can be found in water, sewage, or decomposing organic matter, and in any moist environment. So this is the most important point: moist environment. So as any moist environment can nurture this bacteria, in hospitals particularly, where there is moist environment, where there is moisture and water present, Pseudomonas can easily grow there. And when Pseudomonas is growing in hospitals, it can cause major problems in the hospital in form of hospital acquired infections. So as we all are working in the medical setup, so we should be having some basic knowledge about hospital acquired infection, and which are the important bacteria or microorganisms which can cause common hospital acquired infections. So Pseudomonas is one of the important members of microorganisms which can cause hospital acquired infection. So you might already be knowing what hospital acquired infections are. So hospital acquired infections are simply the infectious diseases acquired in the hospital environment because of the hospital environment. So patient comes for treatment of some particular condition to the hospital, may it be outside patient department, may it be the wards, OTs, or ICUs. So any part of the hospital where patient is getting the treatment, because of that particular environment in OPD, wards, ICU, or OTs, or because of the hospital related procedures, medical procedures, like any procedure like suturing, dressing, or major procedures like operations. So because of the medical procedures, or because of the hospital environment, the patient gets an infection which is from the hospital uh, premises. So this is called as hospital acquired infection. So patient is coming from one condition, coming for the treatment of one condition and getting the infection. So that becomes the second problem. So hospital acquired infection are important to notice and important to look after. So in hospital acquired infections there are many microorganisms which can cause this problem in hospitals. Pseudomonas is one of the important members. As it is resistant to many common antibiotics and many disinfectants or antiseptics used in hospitals, it is very easily uh, able to survive in the hospital environments. As you know, for many infectious diseases, many antibiotics are used, which are used to kill the infection, kill the bacteria causing the infection. But Pseudomonas is resistant, intrinsically resistant or naturally resistant to many of such antibiotics. And in hospital environment, to reduce the bacterial load from the surfaces or instruments or from the skin of the patients, we use disinfectants or antiseptics. So these are actually the agents which reduce the bacterial load, and these are very commonly used in hospital setup. Pseudomonas is resistant to many of the disinfectants and many of the antiseptics used in hospital. That means Pseudomonas can survive even after we have disinfected a particular area, particular instrument or particular surface, and that's why uh, Pseudomonas can cause hospital acquired infections. So nosocomial infection is another term for this hospital acquired infection. So nosocomial infections are nothing but hospital acquired infections. Now the important point is Pseudomonas can even grow in antiseptic solutions. What does that mean? It means that Pseudomonas can gain its nutrition from the chemical present in the antiseptic solution. So this is a big problem, because what we are using to kill the microorganism is actually providing nutrition to Pseudomonas. Not many organisms can do this. Pseudomonas is one of the rare microorganisms which can grow in such chemicals like antiseptic solutions, lotions and ointments, and even some eye drops like antibiotic eye drops. So Pseudomonas can use these chemicals, which are actually toxic to microorganisms. Pseudomonas can use these chemicals to grow. It gains nutrition, and that's why when such infected solutions, infected chemicals, infected lotions and medicines are used for treatment of the patient, this chemical becomes a problem because it can cause Pseudomonas infection in that patient. So here we will see basic morphology of the Pseudomonas, how the bacteria is uh, observed in gram staining, and what are some other basic characteristics. So this bacteria is a rod shaped bacteria. That means it is a bacillus. Bacillus is the singular term and bacilli is the plural term. So rod shaped bacteria are called as bacilli, and spherical or circular shaped bacteria are called as cocci or coccus. So Pseudomonas is not a coccus, it is a bacillus. So plural form of bacillus is bacilli. And in gram staining, which is a very commonly used staining in microbiology labs and for diagnosis of infectious diseases, so that staining uh, can give you different groups of bacteria like gram positive bacteria or gram negative bacteria, out of which Pseudomonas is found to be gram negative bacteria, or we can simply say it is a gram negative bacilli. So that is the basic morphology. It is a rod shaped bacteria and it is gram negative. Then it is non-fermentative. The meaning is it doesn't ferment any sugars. So this is one of the differentiating feature which is used in microbiology for identifying different bacteria, like which bacteria ferments which sugar or which sugars. So like E. coli can ferment lactose, sucrose, glucose or some other kind of sugars, and similarly different bacteria can ferment different kinds of sugars, but Pseudomonas does not ferment any kind of sugar. The third point is it is non-sporing. What does that mean? It does not produce any spores. As you might already be knowing what are spores. Spores are actually the structures of bacteria produced for survival in the harsh conditions. Whenever there is depletion of nutrients, whenever there are adverse conditions faced by microorganism, bacteria particularly, they produce spores. These spores are able to survive in harsh conditions, dry conditions, high temperatures, or in presence of certain chemicals. And after surviving in that condition, when the conditions again become favorable, like nutrients are available, temperature is lowered down, or moisture is available, and the environment becomes suitable for the bacteria to grow, these spores germinate. That means they give rise to bacteria, and the next generations of bacteria are produced. So that is what is a spore in bacteria. But Pseudomonas does not produce any spores. There are certain examples of bacteria which can produce spores, which are Bacillus anthracis or Clostridium. Clostridium is one of the important bacteria which you should be aware of, because Clostridia can cause tetanus-like disease or gas gangrene, which are important for musculoskeletal system. So Clostridium is one of the important example of sporing bacteria or spore forming bacteria. And here Pseudomonas is non-sporing. It does not produce any spores. Then fourth characteristic is it is motile. This is one of the, again, a test to identify different bacteria. We observe the microorganism under microscope and we look for their ability to move, that is whether they are motile or non-motile. The bacteria which are having flagella are able to move, that means they are motile. So Pseudomonas is having the flagella, which is a organ of locomotion in bacteria. So Pseudomonas is motile bacteria. Like Klebsiella is non-motile bacteria, E. coli is motile bacteria. So like that, Pseudomonas is motile bacteria. Now culture. What culture means? Culture means growing the microorganism or growing the bacteria in vitro. In vitro means outside the body. We try to grow the microorganism outside the body. That means in the lab. We try to grow the microorganism in lab. So to grow different bacteria, we use different culture media. Culture media means any chemical composition or any uh, organic composition which is used for growing different types of bacteria. So we will see which culture media can be used for growing Pseudomonas in lab. So basically the Pseudomonas is strict aerobe. That means it cannot grow in absence of oxygen. Oxygen has to be there in environment for the growth of Pseudomonas. So Pseudomonas is strict aerobe. There are certain other bacteria which can grow in absence of oxygen. Even if oxygen is absent from certain environment, some bacteria can grow, like Clostridium. Clostridium is able to grow even in absence of oxygen. So these bacteria are called as anaerobes. But Pseudomonas is different. It is a strict aerobe. Oxygen has to be there. It can grow well on nutrient agar. Nutrient agar is a very simple basic culture medium used in microbiology labs. So Pseudomonas grows on that basic culture medium called as nutrient agar. Certain other nutrient culture media are also there, but simple medium like nutrient agar can also show the growth of Pseudomonas. The growth of Pseudomonas can be observed in wide range of temperature, like 5 degrees Celsius to 42 degrees Celsius, like as low as 5 degrees Celsius and as high as 42 degrees Celsius. But the optimum growth, the best growth or rapid growth is observed at 37 degrees Celsius, because that is the temperature which is seen in normal human body. So best growth rate is observed at 37 degrees Celsius, so it is called as optimum temperature. But at slower rate, it can also grow at 5 degrees Celsius and from 5 degrees Celsius up to 42 degrees Celsius it can survive and grow. And when it is grown in different culture media, the colonies have a certain characteristic, like they are produce they are producing certain kind of smell or aroma. So the Pseudomonas which is able to produce sweet aroma, the smell is sweet, or it can also be earthy odor or musty odor. So earthy smell, whenever there is raining and we can smell the soil, it is a certain kind of smell which is very similar to the smell produced by Pseudomonas. So as we already have seen that Pseudomonas can be present in the environment, it can be present in the water and soil. So whenever there is raining, these chemicals produced by Pseudomonas are released in the air and we can smell those chemicals. So that smell is called as earthy smell, and the same smell can be appreciated when Pseudomonas is grown on certain culture media. So here we are going to see which culture media can be used for growing Pseudomonas. Here we are able to see the growth of Pseudomonas on basic simple culture medium like nutrient agar. You might already have seen the images of nutrient agar in different previous lectures. So actually this is how the nutrient agar looks like. It is a very simple basic culture medium, and it is almost colorless. A slight tinge of yellow color is there, and it is semi opaque or semi transparent. So this is the actual unused nutrient agar plate, and in above two pictures you can see the growth of Pseudomonas. So greenish bluish color is produced by the growth of Pseudomonas. So wherever there is greenish bluish color, that is the growth of Pseudomonas. So Pseudomonas, multiple numerous bacteria, Pseudomonas, are growing on that culture medium. And this is just a simple example how non-pigmented or colorless growth of bacteria can be seen on nutrient agar. So that is different. So this is not growth of Pseudomonas, this is a another bacteria grown on nutrient agar. So this is just for comparison. Only about two pictures are important. This is the Pseudomonas, greenish and bluish colored growth or pigmented type of growth seen on nutrient agar. So Pseudomonas is an bacteria which produces pigment. Pigment means colored chemicals. So these chemicals are bluish greenish in color, as you can see in the photograph. Then this is the growth of Pseudomonas on Meconium agar. On the left side you can see the unused or uninoculated culture media plate of Meconium agar. So this is a pink colored transparent culture medium, and when certain kind of bacteria are grown on this culture medium, certain changes are seen in the culture medium. Like on the right side you can see the culture media plate has been inoculated with two different types of microorganisms. In section A you can see the pink colored round colonies. So these colonies are produced by bacteria. Numerous bacteria produce such typical cluster or typical group arranged in this fashion. So this is called as a colony, bacterial colony, and they have certain characteristics. Like here the colony is pink colored. On the right side in B section you can see colorless colonies. So these are described in microbiology as a non-lactose fermenting colony. As already seen, Pseudomonas does not ferment any sugar, so it produces non-lactose fermenting colony as seen in B section. In A section, it is just for comparison, it is produced by E. coli. E. coli is a lactose fermenting organism. So lactose fermenting organism produces pink colored colonies, and non-lactose fermenting organism produces colorless colonies. So that is seen in B. So A is lactose fermenting, forget about that, it is produced by E. coli, and B is the non-lactose fermenting colony on Meconium agar produced by Pseudomonas. This is just for comparison. So on the left side, uninoculated or unused culture medium can be seen. On the right side you can see the growth of Pseudomonas on Meconium agar. This is one of the selective culture medium which is used specifically for Pseudomonas aeruginosa. So here citrimide agar is the specific or selective culture medium used for Pseudomonas. You don't have to remember all these details, just the basic names, just the basic characteristics you can memorize and you can write in your theories. So just the basic characteristics. So citrimide agar is the selective culture medium used for Pseudomonas. In the upper half you can see the unused culture medium or uninoculated culture medium. It is very similar in appearance to nutrient agar, very similar to nutrient agar. And on the lower half you can see again the pigmented growth of Pseudomonas on citrimide agar. So citrimide agar is the selective culture medium for Pseudomonas. So as already seen, Pseudomonas can produce pigments. Pigments means it produces colored chemicals. There are different types of chemicals. You don't have to remember all these names. I will just name them like pyocyanin, pyoverdine, pyorubrin, pyomelanin. So different kind of color like blue, green or red or black colored colonies can be seen in Pseudomonas growth in culture media. So basically simply saying Pseudomonas is a pigment producing bacteria. These are certain tests done in microbiology lab to identify whether the growth obtained in any culture medium from clinical samples received from the patients is of Pseudomonas or some other bacteria. So like citrate test, like oxidase test, catalase test, indole test, methyl red test, TSI test. These are the different microbiological tests which can be done for identification of bacteria. So these are just two pictures. On the left side you can see the citrate test, positive and negative. On the right side you can see the oxidase test, positive and negative. So Pseudomonas is citrate positive as well as oxidase positive. Now resistance, how these bacteria can survive in different conditions. Why it is important to know in relation to Pseudomonas is we need a Pseudomonas free environment in the hospital, because if in hospital we have not taken care of this Pseudomonas bacteria, they can cause hospital acquired infections. So we should be able to eliminate that bacteria from our environment, from different surfaces, from different objects, from different chemicals. So how to eliminate this bacteria from hospital environments? So here Pseudomonas can be killed at 55 degrees Celsius in 1 hour. So this is a lower temperature. As you can see, 55 degrees Celsius is a low temperature. 55 degrees Celsius will take 1 hour to kill this bacteria. But if we increase the temperature like 60 degrees Celsius, 80 degrees Celsius, 100 degrees Celsius, 120 degrees Celsius, higher the temperature, less will be the time required for killing this bacteria. So increasing the temperature can kill Pseudomonas. Then second point is it can grow in most antiseptics. So we should be aware of the concentration of the antiseptic or chemical composition of antiseptic, and we should know that which chemical is able to kill this Pseudomonas and which is not able to kill the Pseudomonas. It is sensitive to glutaraldehyde and silver salts. So glutaraldehyde is one of the disinfectant used in hospitals, and it is used importantly for endoscopes. Endoscopes are the instruments used for medical investigations. So these are cleaned by glutaraldehyde so that Pseudomonas present over the surface of endoscopes is killed. Then it is intrinsic resistant to many antibiotics. So that point should also be remembered. Like we use different antibiotics for treatment of different infections, and Pseudomonas is able to survive even after the treatment with antibiotics. So there are certain antibiotics which can be used for treatment of Pseudomonas, and specifically those antibiotics have to be used when there is infection by Pseudomonas in the patient, like some cephalosporins or gentamicin or imipenem, carbapenem. Such antibiotics, group of antibiotics are effective against Pseudomonas. So when there is infection by Pseudomonas, we can use member antibiotics from cephalosporins, or we can use gentamicin, or we can use imipenem, like that. Now these are certain characteristics of Pseudomonas which are able to cause problem in the patient in which there is infection by Pseudomonas. So Different chemicals like protease, hemolysins, exotoxins, and lipopolysaccharides, which are able to cause damage, tissue damage, and further tissue necrosis and further problems in the patient. So these are produced by Pseudomonas. Now these are actual medical conditions which are produced by Pseudomonas infection, like UTI, urinary tract infection. Whenever a patient is catheterized, it is done for proper urination in the patient, which are commonly bedridden or cannot move around. In those patients, we insert catheter, Foley's catheter, for urination. So whenever patient is catheterized, Pseudomonas can cause urinary tract infection. Second is, it can cause acute purulent meningitis. Like in hospital, for investigation, lumbar puncture is the procedure which is done for collection of CSF, cerebrospinal fluid. So when this procedure is performed, a needle is inserted in the cerebrospinal space around the brain, and as needle is entering in that space, it can inoculate or infect Pseudomonas in that particular space, and Pseudomonas can cause meningitis. Then post tracheostomy pulmonary infection can be caused. Tracheostomy is one of the important medical procedure done for artificial or proper breathing in the patients in certain conditions. So when tracheostomy is performed in the patient, Pseudomonas can enter from that space inside the lungs and can cause pulmonary infection. Then it can also cause septicemia in immunocompromised individual. Immunocompromised means those individuals which are not able to fight the infection properly. So in those, Pseudomonas can cause bloodstream infections. So it can circulate in blood, multiply in blood, and get lodged in various organs and can cause sepsis. It is also called as septicemia. Then very commonly, Pseudomonas can cause wound infections, and very commonly it can cause burn wound infections. Like in burn wards, you can see different patients which are having these green colored infection over their burn wounds. So that green color is actually produced by the Pseudomonas infection. Pseudomonas also can cause eye infections and infantile diarrhea. Like Pseudomonas is not able to cause diarrhea in adult patients, but it can cause diarrhea in infants, children. So this is just a chart representation of pathogenicity of Pseudomonas. As already said, Pseudomonas is important in hospital acquired infections. Now laboratory diagnosis, how to diagnose the Pseudomonas infection. So we simply have to collect a particular clinical specimen from patient. Suppose in a particular patient, particular condition is suspected to be caused by Pseudomonas, we have to collect a appropriate sample like pus or CSF or blood or the discharge from the wound or skin swabs, simple skin swabs, or urine in case of urinary tract infection. Depending on the condition of the patient, we collect different samples. Like in pulmonary infection, we will collect the sputum, like expectoration from the respiratory tract. So after collection of the specimen, it is sent to the microbiology lab, and in microbiology lab it is subject of microscopy. As already seen, it can be seen as gram negative bacilli in microscopy, and it is then cultured, that is it is inoculated on different culture media like nutrient agar, McConkey agar, blood agar, or in case of Pseudomonas, the selective culture medium is citrimide agar, and then it is subjected to antibiotic susceptibility testing. ABST means antibiotic susceptibility testing. So we test that particular Pseudomonas for different antibiotics, like which antibiotic is able to kill that particular Pseudomonas, so that the same antibiotic can be used for treatment in the patient. So that is all about the Pseudomonas. What important you should know, that it is a gram negative bacillus, it is oxidase positive, it is pigment producing, and it is non sugar fermenting, and it can cause one of the important medical conditions such as hospital acquired infections. Now second section for today's lecture is Vibrio. Vibrio is again very similar in many characteristics to Pseudomonas, as it is gram negative bacilli, it is oxidase positive, and it is motile. But the medical conditions caused by Vibrio are different from Pseudomonas. We will see. This is the electron microscopic picture of the structure of the Vibrio cholerae. As already said, Vibrio cholerae is the important member from the genus Vibrio, and it is having again polar flagella, so which is the organ of locomotion, and that's why Vibrio cholerae is a motile bacterium. So as you can see the morphology in previous picture as well as this picture, it is a slightly curved rod shaped bacteria, or simply speaking it is a comma shaped bacteria. So it is gram negative bacilli, rod shape or slightly curved rod shape, and in gram staining it is gram negative. Then it can show fish in stream appearance in gram staining, like many of the Vibrio bacteria are seen like moving in groups, like they are fish moving in the water streams. So this is just simile, like description of the appearance of Vibrio in gram staining. Then it is actively motile, and the motility is described as darting motility, very fast motility, and that's why it is called as darting motility. So Vibrio is a motile bacteria, and the motility is described as darting motility. Like Pseudomonas, it is non-sporing, it does not produce any spores, and it is non-capsulated. That means it doesn't have any capsule. So we will see which different culture media are used for growing Vibrio. So on the left side you can see the TCBS agar. This is the special culture medium used for Vibrio. Vibrio can also be grown on simple culture media like nutrient agar or blood agar or Makonki agar, but this is the special culture medium used for Vibrio. So on the left side you can see the unused plate of TCBS agar. TCBS is the name given to the culture medium because of its composition, chemical composition. TCBS, thiosulfate, citrate, bile salt, sucrose agar. You might not remember all these names, but just try to remember the name TCBS agar, which is used for Vibrio cholerae. On the right side you can see the yellow colored colonies produced by Vibrio cholera on TCBS agar. So that is a characteristic feature, distinguishing feature of Vibrio cholerae. When it is grown on TCBS agar, it produces yellow colored colonies. Some other culture media which are used for Vibrio cholerae are Venkatraman Ramakrishnan medium or Carey Blair medium or alkaline peptone water. So these are actually the transport media used for transporting the clinical specimen. Generally it is the stool specimen collected from the patient. After collection it is added to one of the culture media like VR culture medium or Carey Blair medium or alkaline peptone water, and then it is transported to the lab. Why these culture media are used for Vibrio? Because these culture media are nourishing for Vibrio specifically. So when transport is done, these culture media uh, do not allow Vibrio in the specimen to die. Actually they provide nutrition, and if Vibrio is present it can survive easily in this culture media. So this is basic classification of Vibrio cholerae. You don't have to remember all these names, just remember that there are different members even in the species of Vibrio cholerae, like Ogawa, Inaba, Hikojima. These are different strains, and these can cause slightly different clinical conditions in patients. So basically you should simply remember the important name as Vibrio cholerae. This is how the clinical condition produced by Vibrio cholerae is seen as. So the disease caused by Vibrio cholerae is cholera, very similar to its name itself. Because it causes cholera, the bacteria has been given the name cholerae. So Vibrio cholerae causes cholera. What is cholera? It is actually a type of diarrhea, and it is also called as rice watery diarrhea because the stool, the motion passed, is in the appearance similar to rice water. What is rice water? When rice is cooked in the pressure cooker or in any particular kitchen vessel, we see such kind of whitish turbid uh, slightly milky watery type fluid. It is called as rice water. So the stool passed by the patient is similar to that type of water, rice water. That's why it is called as rice water diarrhea. So what is rice water diarrhea? It is actually a painless and effortless diarrhea. Patient doesn't feel any pain in the abdomen, and for passing the stools, patient doesn't have to perform any efforts. So it is effortless passing or effortless motion of the stools, and it is painless. No abdominal pain is there, but uh, as it is diarrhea, the frequency of passing the stools is increased, and consistency of the stool is very liquidy. So as you know, commonly the stools are semi-solid, but in patients of diarrhea they are liquid. Particularly in cholera, the diarrhea is called as rice water stool because of the similarity of the stool to rice water. And in certain patients, vomiting and nausea may also be present, and sometimes, very rarely, fever can also be seen in the patients of cholera, but that is a rare possibility. Generally, most common feature of cholera in the patients is rice water stool. So what this condition actually causes in the patient is massive loss of water, as the stools are passed in very higher frequency. Like in say one day, patient may pass stools for 7, 8, 10 or 15 times even. And as the stool is very liquidy, there is loss of water, and many electrolytes in the stool like sodium and bicarbonates are lost due to the action of toxin. Vibrio cholerae produces this toxin, and because of this toxin, uh, there is loss of water and electrolytes in the stool in that particular patients. And because of this loss of water and electrolytes leads to dehydration. So patient is losing the water, so patient faces dehydration. Because of that, hemo concentration occurs, and because of that, anuria means patient is not passing any urine. So there is loss of fluid from the stool, and that's why there is no generation or production of urine in the patient, and that's why patient is not able to pass urine. And patient may also land in hypovolemic shock. That means the volume of blood, circulating blood, or volume of water in the body is decreased, and patient may land in hypovolemic shock. Then muscle cramps due to hypokalemia may also be seen, as there is loss of potassium also in the patient. As you might already be better aware of this condition, hypokalemia, which can cause muscle cramps in the patient. So this is because of the loss of electrolytes in patients of diarrhea or patients of cholera particularly. So muscle cramps due to hypokalemia. Now how to diagnose this condition? So as a patient comes to the hospital with the complaints of diarrhea, rice watery diarrhea, we can suspect that patient might be suffering from cholera. And for that, most obviously the sample collection is the stool sample. So stool sample is collected in a sterile wide mouth container, and then the sample is sent to the lab. If the lab is not located nearby the hospital, and if it is distant, that is certain time gap is anticipated for transport of the sample from hospital to the lab. So in such conditions, the transport media, like previously seen, the VR medium or Cariblair medium or alkaline peptone water, these transport media are used. That is, the stool sample collected from the patient is added to those media, and then the sample is sent to the lab. And that is then again similar, the sample is subjected to microscopy, that is gram staining is performed. As already seen, gram staining, the pseudomonas Vibrio is comma shaped bacteria, and it can show the fish in stream appearance. So in microscopy we can see the comma shaped gram negative bacilli, and culture can also be performed. And in after culture and even before culture, we can see the motility of bacteria. So Vibrio is a dartingly motile bacteria, very fast, very rapidly moving bacteria, so that motility can be appreciated in the stool sample in microscopy, and even after culture when the growth is obtained in different culture media, we can see the motility, darting motility. And the culture media, different culture media like nutrient agar, blood agar, macro agar can be used, and the specific culture medium like TCBS agar, as seen in the previous slide. TCBS agar is the culture medium specific for Vibrio. Then different types of strains like Ogawa, Inaba, Hikojima of Vibrio cholerae are identified by serotyping, and antibiotic susceptibility testing ABST can also be performed to see which antibiotics are effective to kill this bacteria. So those antibiotics are used in the patient also. So ABST test can be done in the lab, and those antibiotics can be used in the patient for the treatment. These are certain, again, chemical or biochemical testings which are helpful for diagnosis or identification of Vibrio cholerae, like cholera red reaction or string test. You don't have to remember the further details of this test. You if 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 you remember just the names, that is more than sufficient. So for Vibrio cholera red reaction and string test is performed. As you can see in the picture, the red colored tube is the positive uh, finding in this test, and it is given by Vibrio cholerae. And on the right side, you can see the positive string test in the suspension of growth of Vibrio cholerae. When such metal loop is inserted and then moved away from that suspension, it produces such kind of string. So it is a string test which is given by Vibrio cholerae. You just remember the names cholera red reaction and string test for identification of Vibrio cholerae. This is again the same picture for appearance of Vibrio cholerae on TCBS agar. So on the left side, uninoculated culture medium, on the right side the inoculated. So you can see the yellow colored growth of Vibrio cholerae on this green colored culture medium. So it is the growth of Vibrio cholerae on TCBS agar. So that is all about this lecture. In case of Vibrio, just remember that it causes a gastrointestinal condition. That means patient acquires the infection from infected food or infected water, and diarrhea is caused in the patient. The diarrhea is particularly rice watery diarrhea, and in similarity to Pseudomonas, Vibrio is again gram negative bacilli or comma shaped bacteria, and it is oxidase positive and it is motile. So these are the basic similarities in Pseudomonas and Vibrio. But Pseudomonas can cause hospital acquired infections, and Vibrio causes the gastrointestinal infection or diarrhea. So thank you for your patient listening. If you have any queries, you can certainly contact in the Department of Microbiology. This is just the simple quote for ending of this lecture. Creating a safe and sterile environment for staff, patients, and facility visitors is not only a necessity, it's a responsibility. This is particularly in relation to Pseudomonas. That is, Pseudomonas can cause hospital acquired infections, and there are certain other bacteria also which can cause hospital acquired infections. So it is the responsibility of hospital staff, that is we people who are working in the medical setup, hospital setup, to take care of the hospital environment so that there won't be any overgrowth of microorganisms or bacteria in the hospital environment, and it won't cause any harm to the patients. So it is reduction of the hospital acquired infections, reduction in the occurrence of hospital acquired infections is the responsibility of the hospital staff. So that is all. Here we end the session. Thank you for your patient listening.